Hi, everybody. Welcome to our April Healthy Aging Speaker Series. First off, I'd like to introduce Dr. Earhart. Dr. Nicole Earhart is the director of the Colorado State University's Columbine Health System Center for Healthy Aging and a full professor in clinical sciences at the College of Veterinary Medicine and Biomedical Sciences at Colorado State University. She holds the Ross M. Wilkins, MD, Limb Preservation Foundation University Chair in Musculoskeletal Biology and Oncology. She's also the director of the Laboratory of Comparative Musculoskeletal Oncology and Traumatology and has been involved in limb preservation research, regenerative medicine, tissue engineering, and cancer research for the last 20 years. She's actively engaged in translational aging research to improve health and lifespan for aging patients, both human and canine, and she focuses on comparative medicine as a powerful tool to bring new therapeutic strategies to slow aging. She holds joint faculty positions in Colorado State University's School of Biomedical Engineering, Cell and Molecular Biology program, and the University of Colorado Gates Regenerative Medicine Center and University of Colorado's Cancer Center. In addition to her leadership roles in many prestigious national and international scientific organizations, such as ACVS, VSSO, VOS, and WVOC, Dr. Earhart was also the first woman to be granted a university-level endowed university chair at Colorado State University. Today, she'll be presenting the TED-style talk we originally scheduled to be presented at the open house event titled Great Danes and Granddads, How a Man's Best Friend Holds the Key to Changing Aging.
Oh, thank you so much, Allie. Hey, thank you all for joining us. I know we had hoped to do this in person, and I hope to see you all in person at the future date that we decide to do the open house when it's safe to do that. But for now, thank you for joining us. We're going to go ahead and try to have these speaker series at least monthly, maybe even more frequently. So keep your eyes peeled for more announcements as we go. So what I wanted to share with you today is how man's best friend does hold the key to changing aging. And if you think about it, it's an interesting thing to, well, let me see if I can get this. There we go. Changing aging has been something that has fascinated people for millennia, essentially. And this is evident in writings and art that goes way back into ancient history. This is one example of a depiction of the Fountain of Youth. This was painted in the year 1500 in Germany. And you can kind of see on the left-hand side the older people coming to the fountain, where then they enter the fountain. And as they progress across the Fountain of Youth, they are restored to a youthful appearance. Their wrinkles are gone. They are more energetic, and they leave the fountain restored and renewed, where they're invited to participate in being dressed in finery, and then this beautiful feast and celebration that occurs. And so this concept that we can change how we age has been a subject of fascination to us for, again, millennia. But of course, as we have come to understand biology and medicine, we've understood the fact that aging seems to be a universal process and seems to be inevitable. And we know this because we can observe it on every level. You know, we can look at someone and we can tell whether they are aged or not just by their appearance. But if we go down deeper into under the skin, we can see it in all levels of ourselves. Here on the organ level, you can see a brain from a younger person versus an older person on the right. In our tissues, this is muscle tissue from a young person versus an older person on the right. In our cells, and even down to the molecules within our cells, we can take a single strand of DNA, look at that DNA, and actually tell how old someone is by specific markers within that DNA, which is fascinating. And of course, as scientists, when we find out these things and we observe these things, the first question, well, how does this work? How do we age? What drives aging in our bodies? And so if we're going to study aging, of course, the first thing we need to do is define aging. And actually, this is a little harder said than done. In fact, if you were to ask, say, 10 different people what their definition of aging was, you probably would get 10 different answers. So for the purposes of this short talk, what I'd like to do is define aging the way we do in science, which is the following: that aging essentially is the result of an accumulation of small amounts of damage, we call it micro damage, that happens to our bodies over the course of our natural daily life. So it's the little things that we don't really notice that accumulate over time. You know, when you breathe in pollution or secondhand smoke, or, you know, wear and tear of our joints when we run for exercise, all of those things, and what they manifest as are things like wrinkles or age-related diseases, things like kidney failure or, you know, decline in our cognitive function. All of those things are part and parcel of accumulation of damage that has caused our cells to not function quite as efficiently as they did when we were younger. So it'd be wonderful if we were like this old classic car, you know, and this car is looking beautiful 50 years or so after its expected lifespan. Why is this even possible? Well, of course, it's because someone has taken the time to look under the hood, find out what's going wrong, find out what parts are needing to be replaced or refurbished, and then slowly but surely refurbishing all of those different parts that would have normally worn out, fixing the chips in the paint, etc. And so this car now is functioning like it's much less old than it is. It's functioning almost perfectly years and decades after its expected lifespan. So what if we were like this? What if we could actually reverse the changes in our cells or repair those accumulated damages so that we change the trajectory of our aging instead of being, you know, what we would expect to be able to function much better for much longer after the expected normal decline in our health and our expected lifespan? And this question has fascinated people for the last many, many years, but we're just starting to get answers to this question. So I'd like to pose two different examples. These are both gentlemen. They are exactly the same age. And yet clearly we see something very different about the man on the left versus the man on the right. The man on the left is clearly extraordinarily fit for his age. He happens to be 82. The man on the right is also 82. And maybe a little bit more typical of people in their 80s, not everyone. He's using a walker for mobility, and perhaps he has some memory loss. Maybe he needs a bunch of medication. But certainly just by appearances, you can see that this man on the right is suffering from more burden of age-related conditions than the man on the left. What is it about the difference between these two people? I mean, clearly what's interesting about them is that they're the same age, and yet they seem very different. And fascinatingly, if we think about people in their eighth decade of life or even later, they tend to fall into two very divergent groups. Interestingly, because there's not a lot of people in between these two groups. So either people are very active into their elderly years, like out on the golf course 5 days a week and living very independently and on very few medications, or they tend to have more burden of disease like the man on the right. Perhaps they need some assistance to walk and are on more medications. And what's fascinating about this age group, and especially in late 80s and early 90s, is there aren't a lot of people in between. So by this time in life, it seems like they're either have gone down a very, what we would say, successful aging pathway or a less successful aging pathway. So what really is the difference between these two different pathways? It turns out that the difference exists at the level of our cells, just as we expected, that accumulated damage starts in motion a number of different processes in our cells that we now know are called the hallmarks of aging. These are the 9 hallmarks of aging that are depicted in this little wheel that we see on the left. And this is the culmination of a huge body of science that has been rapidly progressing over the last 15 years or so. And these hallmarks of aging, or what we would call drivers of aging, are things like our genomic instability, telomere attrition, stem cell exhaustion, etc. But the knowledge of what drives cells to age is really a fascinating subject, because now if we look at this gentleman who we would say is maybe undergone a less successful aging pathway, what we find is that this gentleman has many more accumulated drivers of aging, accumulated hallmarks of aging within his cells than his very fit companion. So on a cellular level, these two people actually are different. This man's cells look quite younger than the man with the walker. So this little discovery is absolutely profound, and it's caused us to be able to think and shift our thinking about aging being an inevitable thing, and maybe something, instead of it being inevitable, that we can modify and change. So when we discover this, of course, scientists get very excited and they go back to the laboratory and they begin to do a whole bunch of experiments on organisms like the ones you see in this little cartoon. And this is depicting a yeast cell, this is a worm, a fruit fly, and a zebrafish. And the reason why scientists love to work on this stuff is because, number one, these creatures have very short lifespans. You can look at a lifespan study in a yeast cell, for example, in a matter of hours. And when you get to zebrafish, it's a matter of weeks. The other beautiful thing about these models of aging is that they have very simple genomes. They're pretty simple organisms. And so if we think about the hallmarks of aging and we think, okay, if we can modify that, if we can start to block some of those drivers of aging, the first place we try to do that is in these very simple creatures. And then if we're really lucky and things go super well, we make this very big leap into a warm-blooded mammal. Now, a mammal is very much orders of magnitude more complex than, for example, a yeast cell. And this model, the mouse, a laboratory mouse, is probably the most common model that we use to make the leap between very simple organisms and a more complex organism like a mammal, to see if what we can do is change the course of aging in something as complex as a mouse. And in fact, this has happened. So we take an old mouse and we give it a medication, and it's a medication that blocks these drivers of aging, or at least some of them, and we see changes that we think are associated with either significant slowing of aging or reversal of aging. And this is the actual experiment that has been done. So these mice on the right-hand side are exactly the same age. But the mouse on the left, this guy, you can see evidence of advanced age. These are old mice, and you can see the hunched back and the loss of hair, sunken eyes, muscle wasting, etc. This mouse is the same age as the mouse on the left, clearly not showing the symptoms of age like this mouse. This mouse was given something to block some of the drivers of aging. In this case, it was rapamycin. And now you see the difference. All of a sudden, this aging pathway that would have ended up both mice looking like the mouse on the left has changed this mouse's pathway. And now this mouse looks not only on the outside to be younger, but if you look on the cellular level, this mouse is indeed on a cellular level younger. Well, this is profound, right? I mean, this is a huge discovery. And when this discovery happened, there was a lot of high-fiving in the lab. There was a huge amount of media about this. Stories were picked up, and talk shows were hosted talking about how scientists had discovered the pill that reverses aging. So you might be wondering, well, when did that happen? Because I must have missed it. Well, it happened about 8 years ago. And so the question is, okay, so this big profound study that we could then reverse aging in mice, where is this? Why has this not happened in people? And this is a huge discovery. And if you're asking that question, that's a very good question. And that story actually happens to be a story that's been told over and over again. In fact, every time that we try to go directly from our laboratory, mouse, for example, or other models of aging, and we try to translate that into people, into applications or medications in people, we end up having to cross this big divide. And we call this divide the translational gap. Basically, what this describes is the gap between these wonderful discoveries that we make in the laboratory and the ability to make that also work in people. And time again, we've failed to translate what we can know we can do in mice and bring that to people. Well, why is this? Of course, you probably have already intuitively understood that mice are not people, right? I mean, mice are nothing like people. Yes, they're a warm-blooded mammal, so they're certainly a lot more like people than a yeast cell, for example. But there's a big difference. Mice live in a laboratory. They're in very controlled environments. Laboratory mice are essentially genetic twins of one another, so they don't have the complexity of different types of people and different lifestyles, choices, and things that accumulate over our lifespan that really influence how we age. And in fact, this is something that's been talked about a lot. And if you look at different media outlets online, you can find titles like the ones I've copied and pasted here. Mice are losing their allure as experimental subjects to study human disease. Mice are great for basic research, but when you start modeling human disease, you run up against major differences. I love this one. Laboratory mice don't eat McDonald's, right? They don't have the same lifestyle choices and things that we do. Judah Folkman, who was a very famous cancer researcher, was famously quoted as saying, hey, if you have cancer and you're a mouse, we can take great care of you. That's not the challenge. The challenge is bringing what we know works in mice and applying that into a human population. And that is where we fail, unfortunately, more often than we are successful. So what we need is a bridge. We need something to get us across this gap. And right now we've been stalled on this side of the gap. Discovery has far outpaced our ability to get across the gap and into people, as you know. So where's that bridge? How can we get there? So let's go back to our two gentlemen that we talked about in the beginning, the very fit 82-year-old and the less successfully aging 82-year-old. We know now three things about these people. We know that this man on the left, on a cellular level, he seems younger. The man on the right, we know, has accumulated far more drivers of aging than the man on the left. And we know that it conceivably, if we could do this in mice, we should be able to do this in people. The same drivers that drive aging in mice also drive things in people. What is missing is, number one, the complexity of a person and all the choices that we make and all the accumulated damage that we occur over our lifespan. And the other big piece that we are missing, that we have no way of knowing in a human population, is when did these two people begin to diverge in their pathways? We know that sometime between the time these two gentlemen were born and the age that they are now, which is 82, they started to diverge. And maybe that divergence was very small in the beginning, only very small amounts of differences could be measured between these two men. But as time went on, that divergent pathway began to get greater and greater apart until we end up at this place where we have two very different individuals. That piece of information is really crucial if we are ever to understand how blocking the drivers of aging can change the aging trajectory to bring this man more toward the trajectory of this man. The problem is that in humans, we don't know when that happens. And that has been a huge barrier to bringing some of those treatments that we talked about working in mice to people. So here are two additional pictures. And what you can see here are two dogs. Obviously, there's a small dog on the left. This is a Jack Russell Terrier, and a large dog, a Great Dane, on the right. Both of these dogs are 9 years old. But clearly there's some very important differences between these dogs. This dog on the left, the Jack Russell Terrier, is clearly an athlete. I think this dog was the national flyball champion in 2012. 9-year-old Jack Russell Terrier, extraordinarily fit and healthy. And on the right, we see a Great Dane who, if to the practice eye anyway, you'll see is exhibiting some signs of advanced age. You can see the arched back, the hunched back, kind of like the mouse we saw a little bit earlier. There's some muscle wasting. And if you were to examine this dog, you might hear a heart murmur, and she might have cancer. This dog, this 9-year-old dog, is a geriatric dog. This dog on the left, the Jack Russell Terrier, is a fit athlete that's in the prime of their life. So those of you who are dog lovers will say, well, Nicole, of course, because Great Danes only live a short time, right? They're a very short-lived breed. And it turns out that's true of all giant breeds, that they live half as long or sometimes 1/3 as long as the small breed, the toy breed dogs. And what's interesting about this is that this is a very consistent finding, that toy breed dogs age more slowly than giant breeds. So in essence, this very fit Jack Russell Terrier is very similar to this 80-year-old gentleman, whereas this Great Dane, who's also 9 years of age, is similar to this 80-year-old gentleman. Both dogs are the same chronologic age, but from a biologic standpoint, this dog is much older and has accumulated more burden of disease. Here's the catch, though, where we don't know when these two people began to differ in how they age. We don't know when they began to diverge. We actually do know this in the dog. The Great Dane begins to age in rapid motion when she becomes an adult. So when these two dogs are puppies, they are aging at exactly the same rate. And as soon as they become mature, all of a sudden this Great Dane begins to age in rapid motion. Well, this is a huge understanding, right? Because now we have an opportunity to intercede at a time when those pathways begin to diverge 1 year later, 5 year later, with some of those blockers of aging, to see if we can influence the pattern of aging and bring the trajectory of this dog's aging process to be more like this dog's aging process. Same with bringing the person from this trajectory to be more like the gentleman that is in better health. Well, that is incredible because we have no way of knowing that in a human, but now we understand that we actually do know when those dogs begin to diverge in their aging process. But not only that, dogs are aging naturally, right? They get all the diseases of aging, but we don't give them. We don't give them arthritis or cancer or whatever. They just naturally get them, just like people. And so because they age naturally alongside of us, they're a far greater mimic of human aging than, for example, a mouse, which is generally we're giving them age-related diseases or genetically modifying them to figure out how to treat that. Not only do they age naturally beside us, but they also are exposed to all the things that cause that micro damage we talked about very early in the talk. They're exposed to things like our lifestyle choices, sedentary lifestyle versus active. They're exposed to the pollutants that we breathe in, our secondhand smoke. They even share much of our same diet and certainly our same water. And also, unlike laboratory mice, which are essentially genetically identical twins to one another, dogs exhibit the complexity in their genetic and epigenetic makeup that is much more similar to humans than a laboratory mouse. So all of a sudden we realize, which really we hadn't before this time, that we have actually this beautiful bridge that has been aging naturally alongside of us all of this time. And that by utilizing that bridge, we believe that we can actually span that gap. Not only can we make older folks age in a more healthy way, but we can make older dogs age in a more healthy way too. So it's a win-win for everyone. So now we know that we can take an old mouse, we can give it a treatment that blocks those drivers of aging, and we can slow aging and make that mouse healthy into old age. We believe that we can take old dogs, do exactly the same thing, and help them be healthy into old age. And finally, using this as the bridge across the translational gap, bringing those treatments to older people and helping them age in a very healthy way. So I want to conclude the talk before we start with questions with a personal story of my own to maybe drive this home. So many of you know that I am a veterinary surgeon by training, and I have been so fortunate to have wonderful mentors in my life who have really helped me understand how to think outside the box, how to look across disciplines to address grand challenges like aging. And the gentleman on the right here is one of my mentors. His name is Don Piermati, and he was what many consider to be the father of orthopedic surgery and veterinary medicine. And I was very lucky to work with him early in my residency, and he became a wonderful mentor and in some ways a father figure to me, and continued to, after he retired, to stay involved in my career and provide advice, and just was very interested in my career path. This picture was taken when he was receiving a Lifetime Achievement Award about 15 years ago. And this picture, unfortunately, was taken about 3 years ago. And this is Don. He developed liver cancer, and it was a terminal case of liver cancer. So he was at home on hospice. And what you don't see in this picture is two little rescue dogs that are curled up next to him there in his bed, keeping him company. And I was able to visit him, and we had a beautiful conversation about a lot of things. And my purpose in visiting that day was to just really thank him for all that he had done and contributed in my life. And we spent a great afternoon laughing about good old times and things. And at one point in the conversation, he got really quiet, and I asked him what was on his mind. He said, it feels too soon to be done. I wish I had more time. It's so wonderful to see how, I can speak into the life of people like yourself and influence. And it's just been such a beautiful journey. And I just wish I had a little bit more time. That conversation has stuck with me ever since, because unbeknownst to me then, he was to pass away only 48 hours later. So I can't help but think, he's right. I wish there was more time. I wish there was more time for all the people that we love and the great minds that have challenged us and caused us to think outside the box. And what I was able to share with him that day was that what he leaves behind is certainly so much more than just his textbooks and his, his works within veterinary medicine. It was the legacy of the people that he trained, like me. But even now, and this is only 3 years ago, I can't help but think that the ending could be changed. Could be changed for a lot of people, knowing what we know now. And, I firmly believe that the creatures that we share this planet with, particularly those that share our homes, hold the key to our health and longevity. And by partnering together and working and reaching across scientific boundaries and medical disciplines, across age groups and societal barriers, that we actually can change the aging and the ending for all of us. So thank you very much for your attention. I'm going to stop sharing my screen and I'll have Allie start with finding out what questions you might have.
Thank you so much, Dr. Earhart, and thank you everyone for sending me some questions. The first one I have is, are dogs sentinels of human health because they share similar exposures and have more genetic variability than lab mice?
That bingo. I don't know who asked that question, but whoever you are, I want to shake your hand because that couldn't have been a better segue. Yes, dogs are wonderful sentinels. And we believe that we'll be able to even show this on cellular levels, that dogs living in the same household as humans will actually, their aging pattern will predict the pattern of aging that's in their human companions as well. And we are putting together an effort to have a biobank that will help us understand those processes in a greater and deeper way. So thank you for that question.
Right. And then the next one is, if you were to design a study looking at aging in humans and dog companions, would it be wise to restrict dog companions to certain breeds or a certain size range?
Yeah, and that another great question. Thank you. So I guess you can look at it in two different ways. I mean, my perspective, I think that restricting it to breeds would be maybe changing, probably getting rid of the variability that we want dogs to mimic. So in other words, if you're within a breed, those dogs are less genetically variable than a human population. And so restricting it to breeds does take out some of the variability that we're counting on to help us mimic human population and aging. On the other hand, saying, okay, well, maybe we should restrict this to large breed dogs because they're the ones that age rapidly and we're most likely to, make an impact in that type of dog that ages rapidly. So I think you can probably, think of the argument from both sides of the coin, and depending on the question you're asking, would probably determine how you would design that, whether you would restrict it to certain breeds or not.
In sort of that same vein, would you think, do you think dogs are a better model than cats?
Oh, cats are great. Thank you for bringing that up. So cats, of course, also live in our homes and share all the same environmental drivers that we talked about the dogs share. The one disadvantage to cats is that they don't have that age difference based on body size. So where we can predict that a large breed dog will age more rapidly than a small breed dog, and that's a powerful thing for us to be able to know, because now we can intercede in that aging pathway at known time points along that divergent pathway, right? That gives us a very powerful look into when an intervention would have the most effect. Cats, unfortunately, we don't have, that same predictable variability because they don't have that obvious body size difference. So they still serve, I think, in many ways as sentinels in a very good way, but maybe is a little bit harder to unravel, healthy aging versus less successful aging in the cat population than it is in the dog population.
Great. And you spoke about treating older animals, people to help them age better. What about prevention?
Prevention, was that the question?
Yep.
Okay. Yeah. So, and so we know that a lot of the lifestyle choices that we make, that we know improve longevity in our health span. So the number of years that we're healthy. are things like lifelong habits of exercise and good sleep patterns and reduction in stress and things like that, even caloric restriction. So, not being overweight and even intermittent fasting across a lot of species, including humans, but almost all species that have been studied for those variables. Those are things that also improve health and longevity in dogs and other species that have been studied. So certainly doing those kinds of things and making lifestyle choices that for your dog or cat that can help with those would be wise. But again, understanding the concept of how could we prevent the rapid aging, say in a large breed dog, that's the kind of thing that we're super interested in, especially with some of these blockers of drivers of aging. And I think that would be an extremely powerful, model to use, as we've already talked about, to understand that mechanism a little bit more deeply. Allie, are you on?
Oh, yep, sorry. I was in chat, sorry. Could you give some examples of molecular targets that are studied at CSU?
Well, yes, many colleagues are doing wonderful research across campus, molecular targets, things like cellular senescence, so specifically specific drivers of senescence and depletion of senescent cells. So those types of drugs are called xenotherapeutics, different mitochondrial energetic pathways. And I probably could list, I don't know, half an hour worth of people doing amazingly beautiful, eloquent research on pathways of aging at CSU, which is one reason why the Center for Healthy Aging exists, to sort of continue to collaborate and develop interdisciplinary science efforts across those disciplines that we've talked about. Because one of the things I think we do really well at a university is we can drill very deeply down into our specific disciplines, into the mechanisms of different aging processes, but sometimes we fail to look across disciplines in a way that would really be helpful to address the bigger complex challenge of aging. And that's what the center exists to do, is to be a hub of interdisciplinary effort to join forces in what has been kind of in some ways siloed work within disciplines. But I think that people are really have realized the value of team science, and certainly we're here to facilitate that. So thanks for the question.
All right. Any other questions that anybody has?
All right. Well, hearing none, we'll let everybody go.
Wait, I'm typing one. I'm typing one. Can I say it out loud instead of typing it, because I'm a slow typer? Okay. Yes. So I was just curious about the social and emotional aspect of aging and how that fits in, because if you're looking at dogs as an aging model, at the same time we're looking at humans aging in the same environment. They're both social creatures, and they both presumably benefit from a relationship with each other. Like, what is the confound there in slowing down the aging process?
Love that question. Thank you. So yes, certainly we know that there are many facets of pet ownership that are beneficial for our aging process, right? And so not only just the fact that by having a pet, we have another creature to socially interact with, which improves loneliness and other things that can be detractors of healthy aging, and our cognitive health and all of that, to the fact that we walk our dogs or we're outside with them, and we can socialize with other pet owners, either actually physically socializing or even virtually socializing. So those pieces of the puzzle are a very important part of how dogs help us, and other companion animals help us age. Even horses and other companion animals clearly are part of the story. So one of the efforts that we're very interested in is looking at and partnering with other people on campus who are experts really at understanding both the mental health and social health of pet ownership in all of its various forms. So I'm extraordinarily thankful for that question, because I think it's still a huge part of the story, and we certainly are interested in continuing to facilitate that type of work and finding ways in which we can not only just look at the physical and cellular changes, but then adding in those aspects as well. That's a very important part of it.
All right, we did just get one more. How may using canines for research affect dementia research or other neurodegenerative diseases?
Love that question. Thank you. So, we know that dogs also experience cognitive decline as they age. And actually, there are now there are very good measures that are validated and repeatable at looking at behavioral changes that are associated with cognitive decline and other actual changes within the brain that mimic dementia and other neurodegenerative disorders in people. So again, thinking about the companion dog and interventions that we could provide that would improve cognitive health. This again is another example of a great, powerful comparative model. So, we can talk about physical health, we can talk about cognitive health, and again, as I think it was Lisa maybe brought up, a mental and social health as well. So, it's one of those opportunities that reaches across so many disciplines, and we're specifically and uniquely positioned at CSU because of the School of Veterinary Medicine to be a leader in this area of cognitive aging studies.
Nicole, could you tell us what some of the blockers or driving agents of aging are, or what is known about it?
Yeah, so the fascinating thing, if we're talking about from a cellular level, if you're talking about the drivers of aging, a lot of the agents that are being studied now are actually repurposed drugs. So drugs that are used for other reasons in the health field, things like metformin, which is a anti-diabetic drug, rapamycin, which is a drug that's used for anti-rejection, dasatinib, which is an anti-cancer drug, even flavonoids and various different food molecules, food-derived molecules, are being looked at. And what's interesting about them is that the doses that are needed to influence the disease that they may have originally been designed for example, an anti-cancer drug or anti-rejection, we are not looking at doses that would have those effects at all. They're very small doses. And interestingly, in some of these molecules, you can take them very intermittently, at least based on the animal research. We think that it may be possible to take them intermittently at specific points in your lifetime, and then be off them for years or months, and then only take them again for a period of time, almost like we're tuning up that cellular damage that we've accumulated over time on a periodic basis. So that's an area that people are actively studying, trying to figure out exactly how and when to use these medicines, not only just within our lifespan, but at what point during our lives, and and can they actually reverse, is there a point where the damage is so widespread that they really won't be effective? And again, not to harp on this too much, but that's where the dog becomes a wonderful model, because there really is no way to do this very precisely in a human population or any really other laboratory animal that has that type of aging divergence that we see in the dog.
Great. If anybody else has any questions, feel free to ask them in the chat, or you can pop on real quick and ask it in person.
Yes, and I will say that keep your eyes and ears peeled for more invitations, because we are going to go ahead and continue the TED Talk series that we had, I should say TED Style Talk, that we had planned for the open house. And then we also are considering doing this on a more frequent basis, depending on your feedback. So please do let us know what you think about this, and we'll make this recording available on our website, www.healthyaging.colostate.edu.
Nicole, any new or surprising insights from your time at Steadman Philippon?
Thank you. You all know me so well. So yes, this is going to be a little deep science, I guess, but I'll try to be as quick as I can, because these are the kinds of conversations I like to have over, I don't know, a bottle of wine or something. But so at the the Stedman Clinic, we had a really interesting study where we were looking at muscle-derived stem cells, so stem cells being the repair cells of many of our tissues. And we were able to harvest stem cells from people who were older versus younger, and then look at what they secreted to look at differences in how those stem cells were able to initiate repair. And as you might expect, the stem cells that were from older folks didn't repair or change the outcome of certain cell types that we were trying to get to understand their function nearly as well as the younger form. But interestingly, when we took those cells and then actually exercised the cells, so outside of the body, putting the cells on a specific machine that actually causes them to be stretched in a way that's similar to exercise, we actually could change that profile so that what they secreted actually did a much better job at causing proliferation and what we believe would be associated with healing effects in even the older cells. So the exercise itself changed how those cells behaved and made them behave more in a way that was like younger cells. So that's the that's the message, is you can exercise and actually preserve your stem cell health for much later in your years than if you're not an exerciser.
How can the community of Northern Colorado assist with your studies?
Oh, love that. Thank you. Well, as we begin to bring more and more online, one of the things that we're interested in is, parallel studies between humans and canine, our canine companions. And so we are always seeking funding to develop clinical trials programs that we could utilize both, human subjects, so people that volunteer for these types of things, whether that's an intervention like exercise intervention or even food intervention versus a medical intervention like some of the things we had talked about earlier. So what I would say is the best way to help us is to stay in touch with us and stay abreast of the various different clinical trials that we will have on our website. And if you're a dog owner or you're a person that's interested in participating in human clinical trials, please reach out, because we probably have opportunities for you to be involved.
And everyone's welcome to email me. It's ali.murphy@colostate.edu if you have any questions about it or want to make sure I got you down on my list.
All right, everyone, please feel free to reach out. My email is there by my picture, below my picture, but you're always welcome just to, search me on Google. I'm sure we'll be able to connect, and I look forward to chatting with you and seeing you all again at our next installment of our Healthy Aging Speaker Series.